Yeah, good evening to everyone. Today we will discuss about to the chromosomes, how to chromosome use, and these are things we will discuss about the topic. Now we will discuss about the topic name is called chromosomes. Chromosomes are Chromosomes are useful for the all genetic information for the other genetic information for one generation to another generation. So here the chromosomes mostly use for the another importance of this one, that is called the chromosomal number. In this one, the chromosomes are also used for the chromosome number and how to use the chromosome. These are things are useful for the chromosome systems. And here the chromosome number mostly constant in all importance of that one, constant that the constant number is present in all organisms. But here this one is the chromosome. The chromosome is the important characteristic features of the all genetic engineering, and this one the the cell organelles and cellular components all are too important for the chromosomes. And here the chromosome number mostly constant in one species to another species. In this one, one second, capital G C 5VF in this chromosomes used for the all organisms. And here the chromosome mainly important for the other systems also. I'll share my PPT. One second. Hello. And in this one, the chromosome mostly used for the where is my screen? Chromosomes. Here, the chromosome number and chromosomes also used for the genetic information from one generation to another generation. One generation to another generation, the chromosomes also used for the constant number is there, and chromosome the structure of chromosomes in the nucleus, the chromosomes also most important. The nucleus is the chromosomes also most important characteristic features of the chromosomes. Here in this one, the chromosomes occur the mainly in a human being is the 24 23 pairs. But another thing, another chromosome. Hmm. This number mostly important for the all organisms, and the chromosome is mostly used for the genetic information for one generation to another generation. These are this one is used for the structure of chromosomes. In between one chromat in the center of the centriole are there, and two chromatids also present. And in this chromosome, in this one, the chromosome also constant number. In the nucleus, the chromatids are present. Chromatids, two chromatids are in the centromere is there, and two chromatids are present in this one. These centromere and chromatids both are the present in the centromere of the chromosome. And here these chromosomes. of the nucleus of each cell, the nucleus of the each cell to produce the chromosomes. And another thing is, one second, what happened? This one is mostly constant number is there. In this constant number and the nucleus of each cell, the DNA molecules, the DNA molecules is prolonged. That one is prolonged, packaged into their packaged into their of the nucleus. In this process, the chromosomes also this one is also you can observe the inside the nucleus also. Here first discovered the chromosome is the Starsberger scientist in 1950s, 1875. The chromosomes first described the Strasburger scientist in 1875. The term chromosome was coined by Waldeyer scientist. The first chromosome was coined by the Waldeyer scientist. This Waldeyer and another in 1888. Each chromosome is made up of DNA. Each and every one chromosome too made up of the DNA. And this chromosome structure this chromosome structure and each chromosomes found into the DNA structure and DNA particles all are the present in the chromosomes. But here the chromosome number is a constant into the other nuclear membrane. Other nuclear membrane. One second, I'll share my PPT here only. Yeah, now it's okay, ma. My voice. Yes, now okay. Now okay, my voice. Minaj. Now it's okay. Yeah, I will. I am not to share the PPT. Just I'll explain, ma. My system is problem is that today we will discuss about to the chromosome. How what is the chromosome? How to use the chromosome? These are things we will discuss about today. Chromosome definition and history. Features of the eukaryotic chromosomes, chromosome size, shape and number. Next one is the chemical composition of chromosomes, chromosome structure. Here the introduction and history of the chromosome. Main important of the chromosome. Chromosomes are rod shape, rod shape, rod shaped dark stained particles is called the chromosomes. Dark stained bodies. These dark stained bodies are called seen during the which stage you can observe the chromosome. The chromosome you can when the interphase, that is the metaphasic structure is there, no, in that metaphasic structure the chromosomes you can observe in the mitotic time. These mitotic chromosomes, the scientist name is called the Strasburger scientist. The Strasburger scientist to explain to the chromosome discovered in 1875. In 1875, the scientist name is called Strasburger. This Strasburger to explain the chromosome. And another one, the chromosome, the chromosome is called the, that one name, the term is the another scientist Waldeyer. Strasburger to explain the chromosomes discovered in 1875. The term chromosome was coined by, the term of the chromosome was coined by the Waldeyer scientist. Waldeyer in the 1888, the year of chromosome 1818 by. What is the chromosome? These chromosomes are called term initiated as Latin word chroma meaning color, zoma meaning body. That is called the color bodies. The name is called color body. The features chromosomes are the best visible when you can observe to the metaphasic structure. When you can observe the chromosome, when the metaphasic structures are there, in that time you can observe the chromosomes. These chromosomes bears genes in linear fashion, and chromosome vary in size, vary in size and shape, number, different species of plants and animals, different species of plants and animals. Chromosome have property of the self duplication and mutations. Mutations. Chromosome are composed of the DNA, RNA and proteins. This is the chromosome. Just what is the introduction of the chromosome? And next one is called chromomeres. Chromomeres meaning the chromosome size. Size of chromosomes is called that size of the chromosome meaning that one mean that is the chromosome size is measured at only for the meiotic phase. Which phase you can observe? Where the meiotic phase is there, in the meiotic metaphase genera measured in length and diameter of the chromosome. Plants usually have longer chromosome than animals. What is that one? Where the longest chromosome you can observe? Plants only. Plants only. Longest chromosomes are present in the all plants compared to compared to animals. Another one, pure chromosomes. The plant chromosomes
are generally 0.8 to 7 millimicron in length, whereas the animal chromosomes are there. In that animal chromosomes, the animal chromosomes size, that one is a 0.5, that is 0.5 to 4 millimicrons. 4 millimicrons chromosomes are observed in the animals. That is the length of the chromosome. What is that one? Length of the chromosomes. That is the length of the chromosomes. Coming to the next one is the chromosome shape. That is called the chromosome. That is the chromosome shape, and the chromosome number of chromosomes in a given species, generally constant number is present. This is most important. Always the chromosome number is constant, where you can take any plant organism or any animal, any living organism, the chromosome number always constant. All the members of the species ordinarily, that is called the ordinarily, that is the how defined, generally a constant stomatic and gametophytic chromosome number. This one, the stomatic and chromosome number, it is the number of chromosome found in stomatic cell of a species. See, this is a haploid and diploid number. And the second one is the triploid and tetraploid numbers also present. Tetraploid numbers also present in this one. The shape of chromosomes. Here, the shape of chromosomes also most important. Chromosome is generally determined by the position of centromere. The chromosome generally exists in three different shape: rod shape, J shape, and V shape. Different shapes are also present in this one. Chromosome number. The number of chromosomes also, each and every species define number and constant somatic and genetic chromosome numbers. Somatic chromosome number is number of chromosome found in somatic cell, while genetic chromosome number is number of chromosome found in the gametic cells. The somatic chromosomes are always diploid and are depends as 2n stage, where the genetic chromosomes were always haploid number and denote as, you can write down n number. In human being, somatic chromosomes name is called XX and XY, where the genetic chromosome number is called X and Y. This is a number chromosome. Somatic chromosome number, some common plants and animals in human being. That is the Homo sapiens. That is about name of the scientific name. The somatic cell, 46 chromosomes are. In gametes, male gamete 23 pair, female gamete 23 pair. Oryza sativa, that is called the rice. 24 stomatic chromosome, 12 male gamete, 12 female gamete. Next one, Rattus norvegus, rat. In carrot, Darkus carota. Darkus carota in carrot, 20 chromosomes somatic chromosome 20. Allium cepha, anion, 16 chromosomes. Next one, Ziamase, 20 chromosomes. Next, housefly, bread mold, neurospora carpus, 14. This is the number of the chromosome. Number is the stomatic chromosome number and gametophytic chromosome number. Different species, different numbers are present. Next one is that one. Next one, the chromosomes are mostly, the structure is the very important. The structure of chromosomes also most important. The structure is the first one. Chromosome consists of 7 parts. In the structure, how many parts are there? Total 7 parts. One is a centromere, second one is a chromatid, third one secondary constructions and satellite, fourth one is the telomere, fifth one is the chromosome, chromonimeta, and sixth one is the chromonema and matrix. Chromonema and matrix are present in the chromosomes. Here, in this one, the chromosomes are the structure is most important chromatids, and these are centromere. Centromere meaning it is a localized. Centromere is called localized region of the chromosome with which the fiber attached is known as centromere or primary constructions, or this one is the kinetochore. Chromatid. Next one of the two distinct longitudinal subunit of chromosome is called as chromatid. These chromatids are two types. One is the sister chromatid. The second one is called the non-sister chromatids. This is the chromatid structure. Next one, secondary construction. Small chromosomes exhibit secondary construction. In addition to primary construction, and the chromosomal region between telomere is called satellite chromosomes. These chromosomes mostly mostly chromosome number is constant. No, this one is the species. This number is constant, but in this one the chromosomic number, that is called the mostly important characteristic feature of the chromosomes. So this chromosome, that one is called the chromosomic number, chromosomes, all are the V shape. You can observe the shape. That one is called that one second. That one is called the you can observe that name is called TC5. One second. This one is the this one chromosome is the most important. Another thing, the chromosome number. Number and constant number is the most important. Here you can observe the downloads and chromosome accepts. These are things are mostly important for the chromosomes. Another thing is the number of chromosomes. You can observe V shape, L shape, and different structures are there. The different structures of the chromosomes also useful for the all the things. Another one is called these chromosomes. Next one is called the chromatid. One of the two distinct longitudinal subunit of chromosomes is called chromatid. Chromatid are two types. One is a sister chromatid, second one is the non-sister chromatid. Next part is the chromosome secondary constructions. Secondary constructions, some chromatids exhibit the secondary constructions. These secondary constructions, in addition primary construction, the chromosomal region between the telomere is called the satellite or terpent. The chromosome having satellite is called satellite chromosome. That name is called SAT chromosome. The two ends of chromosomes are called as telomeres. Telomere are highly stable and they do not fuse or unite with the telomere of the other chromosomes. The chromomere of some of the species show small bead-like structures are called. That name is called chromomere. The structure of chromomere in chromosome is constant. The chromomere are thread-like coiled structure found in the all chromosomes or chromatid is known as chromonema. That is called sometimes to called chromonemata also. That name is chromonemata. Matrix. It is a fluid part with the chromonemata or embedded. It is called as matrix. That is called the matrix. Another name is called chemical composition. Now the chromosomes are the chemical compositions. Chemical composition, chromosome nucleoproteins. Chemical composition of the chromosomes is called nucleoproteins. These nucleoproteins is called, that name is called, nucleoprotein name is called, nature in the R or they are compound two types, RNA, DNA and proteins. These general chromosome consist of the DNA percentage 30 to 40% and proteins 50 to 65% proteins are there. 0.5 to 10% RNA is present. The amount of DNA present in somatic cell is constant. The DNA content of genetic cell, gametic cell is half of that somatic cell. The DNA of chromosomes is of two types, unique DNA and repetitive DNA. Two types of DNAs are present, unique DNA and second one is the repetitive DNA. Unique DNA is called uh, unique DNA is called Unique DNA is called. Those DNAs are importance for the chromosomes. That is not that is no, that is a unique type DNA is the most important for the another things. This one is DNA main important for the our genetic engineering and gene structures. These are things also most important for the DNA. And in this one, the DNA structure and the percentage of DNA also depending upon the chromosomes. And here another one, the DNA of the content gametophytic cell is of that somatic cell. DNA, unique DNA consists of those DNA sequence which are present single copy per genome and unique in nature. This unique DNA is also known as non-receptive DNA, code for the which require large quantity of the cell for storage of proteins. For storage of the proteins. Another one, repetitive DNA. Repetitive DNA is called, this DNA consists of DNA molecules or base sequence, which are these base sequence, which are base sequence, which are these are the main base sequence. Base sequence is the most important. Repetitive, which are few several hundred base pairs, that is called BP. Along are present to several million copies of a genome. The human genome contain 30% repetitive DNAs. Repetitive DNA is further divided into highly repetitive DNA and moderately repetitive DNA. This is the repetitive DNA. Coming to the next, RNA. RNA purified chromatin. 10 to 15% RNA is present total DNA. This RNA associated with chromosome is messenger RNA, transfer, three types of RNAs: mRNA, tRNA, rRNA. mRNA meaning messenger RNA, tRNA transfer RNA, rRNA is called the ribosomal RNA. Next one, proteins. Proteins associated with chromosome is classified two main groups. Histone protein, that is called basic proteins. Second one, non-histone proteins. Two types of proteins are there: histone proteins and non-histone proteins. Both are present. Histone proteins, histone proteins are called, histone proteins constitute of the, non-histone proteins are acidic in nature. Non-histones are the acidic in nature, and histone proteins are basic in nature or basic amino acids. Histone proteins constitute about 80% of the total chromosomal protein. They are present in an almost these are the 1:1 ratio with DNA. Five fraction of histones are present, like 1H1, 1H2B, and 2H2B, 2H23, 2H3, 2H4, like this. These are the histones. Non-histones proteins make up of 20% of the total protein mass. These content of non-histone protein is different from species to species. It is not particular. This one is the different from the species to species. Non-histone protein includes many important enzymes like DNA and RNA polymerase. This is the non-histone. According to the relative position of centromere, chromosome are divided into four types. One is the metacentric, second one submetacentric, third one acronetic, fourth one telocentric. Centromere. This one is the centromere position. Okay? The localization of centromere, the chromosomes is different. Here in this one, chromosome is a karyotype. What is a karyotype? Complete set of chromosome in a species or in a individual organism is known as karyotype. What is the meaning of karyotype? Complete set of chromosome. in a species, complete set of chromosome in a species or in an individual organism is known as the karyotype. The basic number of chromosome in the somatic cell of an individual or a species is called somatic number and is designed to n. That's where the human chromosome 2n is equal to 46. Another important character for chromosomes, that is ideogram. Ideogram meaning the diagrammatic representation of chromosome morphological characteristic of a species or a population is known as ideogram. The meaning of ideogram: ideogram represents of chromosome morphology chromosome characteristic of a species or a population is known as, that is called the ideogram. That name is called the ideogram. Ideogram, when you can observe the ideogram, the structure of ideogram, that one is that name is called the ideogram, is called the main important of the size of chromosome. What are the abbreviations? Chromosomal abbreviations is the structural changes, numerical changes, that is called the chromosomal abbreviations. Structural chromosomes again intrachromosomal, interchromosomal. This is the deletions, duplications, inversion. Interchromosomal is a translocation. Numerical chromosome is called the aneuploids, euploids. That is called the numerical chromosomes. What are the translocation? What is the meaning of translocation? It includes all type of unilateral or bilateral transfer of chromosome segments from one chromosome to another non-homologous chromosome. Here, the genetic significance of translocation is the genes from one chromosome to another chromosome. One chromosome to another chromosomes. This here, one generation to another generation, one generation to another generation, the chromosomes are important for the genetic significance of that one. Genetic significance of the translocation is that genes from one chromosome are transferred to the another chromosome. Next one, reciprocal translocation. When the pieces of two non-homologous chromosomes are interchanged without any net loss of genetic material, the event is referred to as a reciprocal translocation. Before translocation and after translocations. The here you can observe the three orientation: metaphase 1, alternate, adjacent 1, adjacent 2. In this one is the metachromosomes abbreviations. And in these chromosomes, main important of the chromosome bonding and chromosome binding patterns. Many binding patterns also there. Here main important is the karyotype. Karyotype may be defined as the study of chromosome, morphologically chromosome, complete in the form of the size of chromosome, shape, position, primary construction, centromere, secondary construction, satellite chromosome, defined individual of the some chromosomes, and any other additional features. The karyotype highlights closely or distantly related species based on the simultaneously or dissimilarly of the karyotype. For example, a group of species resemble each other in the number, size, and form of their chromosome. Asymmetrically, the karyotype may be defined as the huge difference between the largest and smallest chromosome as well as the number as well as the number also important as well as the number also most important for these chromosomes. Another thing, all chromosome asymmetrically, for example, a group of species resemble each other in the number, size and form of their chromosome. Asymmetrically, karyotype may be defined as the huge difference between the largest and smallest chromosome as well as less number of metacentric chromosome in a chromosomic complete. The principles ways, the principal ways in which karyotype differ from each other are basic chromosome number from and relative size different chromosome of the same set, number and size of satellite related those position of the chromosome which form nucleoli and secondary constructions. NOR region chromosomes, also absolute size of chromosomes, distribution of material which differ different staining properties, that is euchromatin and heterochromatin. Binding the techniques, this is the technique for the identification of chromosomes. Abnormalities in the chromosome complement. The chromosome definition depends on their morphological characters such as the relative length, arm ratio, presence and absence of secondary metabolism, secondary constructions on the chromosome arm. Therefore, it is an additional and useful tool for the identification of individual chromosome within the chromosomic complete. Further, it could be used for identification of chromosome segments that predominantly consist of the guanine, cytosine, adenine, thymine rich region constitute of the heterochromosomes. These binding techniques of the chromosome, the binding techniques which involves denaturation of the DNA, DNA followed by the slow permits identification of constitute exochromatin. So constitute exochromatin as it mainly consists of a repetitive DNA. On banded chromosome, darkly stained or brightly flavored transfer bonds. That is the alternative with the highly sanitized or less fluorescence negative bodies are present, where the bands are complete. The bands as constant, reproducible constant and reproducible are each pair of the homologous chromosome. Binding technique also revealed in this excessive genetic polymerization manufacture. This is the binding materials of the chromosome. And these code systems also most important chromosome band, that is a quarisonic bonds, constructive heterochromosomes are there, gemagic bondings are there. These are the chromosomes bondings and chromosome structures. This is the chromosomes non-homologous and homologous. All chromosomes also sister chromosomes, and these are things are the most important for the chromosomal structure. Another very important, the chromosomes, if you, this one is mostly in the chromosomes, these are the in it. And here, structure, each chromosome different into three parts are the pellicle, matrix and chromonemata. And here, most important for the telomeres also very important. These are the end part of the chromosomes, exhibit the physiological differentiation and polarity also present. Each and every extremely of chromosome due to the polarity prevent the chromosomal segments to be fused with it. The chromosomal ends are known as the telomere. If a chromosome breaks, the broken end can fuse with the each other due to lack of telomere. The autosomes are sex chromosomes also present. Human chromosome, two sets of chromosome. One is the autosome, the second one is the sex chromosome. Genetic traits are linked to the sex of the person, are passed on through the sex chromosome. The rest of genetic information is present in the autosomes. Humans have 23 pairs of chromosome in their cell, in which 22 pairs are autosomes. Total 23 pairs. How many is autosomes? 22 pairs are autosomes and one pair is sex chromosome, making a total 46 chromosome in each cell. On the basis number, monocentric only one centromere is present, dicentric two centromeres are present, polycentric with more than two centromere, and sometimes acentric also. Without centromere, such chromosome represent fleshly broken segment of chromosome which do not survive for long time. This one is do not survive for the long time. On the basis of location of centromere, chromosome divided in two part. One is the peak and Q arm. Two arms are present, which consist of constrictions point is called the centromere in the middle. The centromere can be located in different positions, and this form of basis of four different classes: metacentric, submetacentric, acrocentric, telocentric. Centromere is in middle, mapping P and Q arm are compatibly same length. That is the meta. Submeta centromere off center leading to the short P relative to the QP. That is a submeta. Acrocentric. Acrocentric centromere severally offset from center, leading too much of the short P arm. Telocentric centromere found at end of chromosome, meaning number of Q arm exists of this one. What is the function of the chromosome? The number of chromosome is constant for a particular species. Therefore, these are the great importance in the determination of the polygyny and taxonomy of the species. Genetic code storage also. Chromosomes contains the genetic material that is required by the organism to develop and grow. The DNA molecules are made of chain of units is called genes. Genes are those section of the DNA which code for specific protein required by the cell for its proper functioning. The sex determination. Humans have 23 pairs of chromosomes, out of which one pair is the sex chromosome. Female have two X chromosome and male have one X and one Y chromosome. The sex of the child is determined by the chromosome passed only one Y chromosome that determined whether passed whether down by the male. If X chromosome is passed out XY chromosome, the child will be female. That is female, and that is the child will be female. If Y chromosome is passed male, child is developed. The control of the cell division also, this one is controlled. How to control? Chromosome check successfully division of the cells during the process of meiosis. The chromosomes of the parent cell ensure that the current information is passed on the daughter cell required by the cell to grow the develop correctly. Formation of proteins and storage. The chromosome direct the sequence of the proteins from formed in the body and also maintain order of DNA. The proteins also stored in the coiled structure of chromosome. These proteins bound to the DNA helps in proper package of the DNA. This one is the total chromosomes importance, sorry, functions. These are the functions of the chromosomes. Here prokaryotic chromosomes also different, eukaryotic chromosomes also different. Prokaryotic chromosomes, here genomes are simple and the organization of DNA is also different. Genomes also contained single chromosome which are usually circular DNA molecule. Here DNA is associated with protein but no histones are present. No histones are present. DNA packing DNA is different form and is not well understood. Variation in genome length bearing gene and RNA viruses having small genome also. This is the prokaryotic chromosome. Eukaryotic chromosome, genomes are composed of multiple chromosomes, each chromosome containing a linear molecule of DNA. Histones are also unique features of eukaryotic cell. The packing DNA is double histosons. Next one is the chemical composition. Chromosomes of thin chromatid thread-like structures. This is a chromosome fibers and interface chromatid consists of a 30 to 40% DNA, 50 to 65% protein, 1 to 10% RNA is found that one. In metaphase, the chromosome contain 15 to 20% DNA, 10 to 15% RNA, 65 to 17.5% protein. DNA consists of nucleotides, while the primary protein component is histones. Histones strongly attracted to the negatively charged phase group of the DNA. There are five major types of histones. Thousands of other types of proteins involved in DNA replication and gene expressions. The degree of DNA, the degree of which DNA is consolidated to expressed as in packing ratio also. Packing ratio meaning the length of DNA divided by the length into which is the packaged. That is the packing DNA. Next one, the achieve the overall packing ratio. DNA is not packaged directly in the final structure of chromatin. Chromatin is composed of repeating 200 base pair unit which were called nucleosome. Nucleosome was first discovered by scientist Korenberg in 1974. Here 146 base pairs DNA wrapped 1.75 times around a stone core. One molecule each one bound in the DNA either and exit the nucleosome particle. This form a chromatin subunit known as chromatosome. Consists of 166 base pairs of DNA. The stone core act as a magnetic form of the coiling of DNA. Nucleosomes also are coiled for supercoiled also present. This one is the chromosomes and these are the functions and eukaryotic prokaryotic chromosomes. Reason of the chromosome. One is the euchromatin, heterochromatin. Several different chemical techniques are used to identify certain chromosomal region by staining them so that they form chromosomal bands. There are two types of bands. One is a euchromatin band, second one is a prochromatin band. Light staining region is called euchromatin. Very light staining. Light packed from the chromatin is often under active transcription, found in both cells with nuclei and cells without nuclei. Heterochromatin, darkly stained, highly tightly coiled, darkly stained, tightly coiled throughout the cell cycle, and that is so far the most part genetically inactive. Next, constitutive heterochromatin. It is fixed and irreversible in the form of function. It does not resort to the chromatin stage. This chromosome 196, Y chromosome 1916 and the Y chromosome contain region of the constitutive heterochromatin. Sometimes facultative chromosomes are heterochromatin. It has faculty to return the normal euchromatin state. Consists of euchromatin that takes on the staining and compactness characteristic of the heterochromatin during some phase of development. The inactive X chromosome is made up of facultative heterochromatin. You know, types autosomes, allosomes, and meta types low metacentric, submeta. These are all the types of the chromosomes. And diffuse of chromosomes. One second. Hello. Diffusion of chromosomes, also known as holocentric chromosome. The entire length of chromosome act as a centromere, and homologous chromosome, non-sister, and another important also special types of chromosomes also there. The special types of chromosome, polytene chromosome and polytene chromosome, and that one is called the next one is the Lambresh chromosome. Next one B chromosome, holokinetic chromosome. Number of chromosomes, size, karyotype, and functions also there. These are the total topic from the chromosome. Okay. Now this topic is completed. Miraz, it's okay. Did you understood? Today my sharing is some problem is there. That's why I am not to share. Did you
Yes,
understand?
ma'am, understood very well.
Yeah, thank you. My laptop is not sharing today. What problem? I don't know. Okay. You, Minas, take my number. If any doubt, you can ask. Write down my number 8143.
1 second, ma'am. Yes, ma'am, now.
8143. 81.
Yes,
8143.
8143.
Again 81.
Yes,
2421.
8143 812421.
2421.
8143 812421.
Yes. Where did you stay, Minas?
Here, ma'am, Medipatnam.
Okay, no problem. I'm work from the Reddy Women's College. If any time you can ask, I'll explain to the doubts. Okay?
Okay, ma'am, okay.
Yeah.
Thank you, ma'am.
Okay, Ra. Okay. Bye, ma.
Bye, ma'am.
Bye, bye. Shall I close, ma?
Yes, ma'am.
Okay, Minas. Bye.